we still, I believe, underestimate the complexity of the human nature. The world around us is not a friendly one. And I mean, we need to have a very, very complex system to constantly fight that back in order to survive.
We just had actually in my town 6 cases of meningitis, and that's come out of nowhere.
2 people have died, one university student and the other understood to be a sixth form pupil, after an outbreak of a rare form of meningococcal disease.
Unfortunately, the way we live over the last couple of years probably has led to the fact that we do not train our immune system enough. And in other cases, have led to the reoccurrence of some viruses. Many of the drugs out there, the TNF blockers, the IL-6 receptor blockers, great drugs, no question. But they get to a certain level of efficacy, and then they hit the ceiling. That's it. We have a strong belief that the key here might really be a combination. It's the monopathways which get you to a certain level of efficacy. But if you really want to get to the next level, you need to hit more than one.
So if everything goes right, what could this company become?
We want to be the IRhom company. So whenever, from a therapeutic perspective, one talks of things about IRhom, in particular IRhom2, it needs to be associated with SciRhom.
I mean, in the end, as long as you keep in mind that you want to help patients to better deal with their disease, or in the best way, if possible, even cure the disease. That should be the ultimate goal always. But
did you know that 97% of our listeners are not subscribed to the Leading Beyond the Lab podcast channel? Be sure to hit subscribe to make sure that new episodes with the guests and companies that we're talking to land straight into your feed. Thank you.
Hello and welcome to Leading Beyond the Lab. Today I have the pleasure of being joined by Poth and Jens Ruhe, CEO and COO of SciRhom, a company developing a new approach to treating autoimmune diseases by targeting IRhom2. For years, this field has focused on blocking individual inflammatory pathways like TNF and IL-6, but outcomes in recent years have plateaued, with many patients still not reaching remission. What I love about SciRhom is that they're taking a different route, targeting IRhom2 to control multiple key drivers of inflammation upstream, with the potential to go beyond that ceiling of efficacy. The underlying science was driven by Jens and the rest of the founding team, with Jan joining later on as CEO to help turn this into a drug development company. Today, I'm really excited to talk about the science, their journey from discovery to development, and what it takes to turn this into a real therapeutic. Jens, thank you so much for joining me on the podcast today.
thanks for having us.
Our pleasure.
So, Jens, I'd love to start by delving into your experience. What was the reason when you founded SciRhom that you thought IRhom2 could be targeted differently this time?
Well, the idea was to have to find new way of targeting TACE, which is the molecule that is being regulated and influenced by IRhom2. So the idea of founding a company on that basically came from one of our co-founders, Carl Blobel, who discovered IRhom2 and just a couple of years before. And TACE used to be a very hot target in the past. So many big companies, pharma companies, tried to develop TACE inhibitors, which all failed for certain reasons. And IRhom2 was discovered as a critical regulator, and that offered the opportunity to target TACE in a different way. And we found that extremely exciting because it was a novel approach. It was a new way of targeting IRhom2, of targeting TACE, which failed before. And we were very optimistic that this is the key of targeting TACE.
What was it that went wrong previously that you felt like you could do things differently this time?
Well, the inhibitors that were developed by different companies before targeted TACE itself. And one needs to know that TACE is a very central protein that is involved in the regulation of multiple pathways, many pathways that are involved in disease, but also importantly, also pathways that are critical for normal physiological processes. And all those inhibitors that were developed targeted TACE globally, which in essence means that you may have the benefit of targeting those pathways that drive disease, but at the same time, you would also target pathways that are important for normal physiology. So, and in essence, that broad interference with TACE, basically the global interference with TACE, led to side effects in the clinical trials and caused them to be discontinued. And now IRhom2 offered the possibility to target TACE in a more selected way. And that is something that was, well, basically the key to readdress TACE.
And you were working at the time. So what was the eureka moment when the spark came, where you decided to set the business up?
Well, there's a couple of facets to that. So the first one was that the company that I was with before, which I also co-founded, was taken over by a big pharma company and then had to be closed down for several reasons. So which actually gave us the time to think about something new.
You need that sometimes, right?
And also at that time, so the target IRhom2 became more and more popular. So there was a dramatic increase in scientific publications. So everybody, or many people, came across that molecule and realized, okay, that's a very important player and is probably a good target. And we had a close interaction with Carl Blobel, who, as I mentioned, was the one who discovered the protein. And
he's a scientist.
Yeah, he's a professor at the Hospital for Special Surgery in New York. And he discovered the IRhom's, and he discovered the important function of those molecules. And due to the close interaction that we have had with him due to the previous scientific work, we had access to his thoughts, to his data. And that's a very funny thing, because at that time when we started the company, the entire scientific community believed that IRhom2 is not druggable, because everybody thought at that time that this molecule is located within the cells, not on the cell surface. So, however, if you want to develop an antibody, that molecule somehow needs to be accessible. It needs to be on the cell surface. And it was only Carl who, on the basis of his studies, on his experiments, concluded, in addition to the important intracellular function, there must be a cell surface associated function. And he conducted a couple of experiments that provided strong evidence that this molecule indeed is located on the cell surface. And since Carl is a fantastic scientist, we believed in his data, we believed in him, and we thought we need to try to see if it's possible to generate antibodies against that molecule, because it's such an exciting opportunity to get a hold on, or to address TACE a different way. Well, I mean, it was, of course, a challenge. Most of our former colleagues thought we are crazy because there were no tools. Everybody thought it's an intracellular target. And why do you want to generate antibodies? Try to generate antibodies against that molecule. But, well, it was the science by Carl. It was the science that was published in the literature, which we thought we must give it a try.
A number of companies had already targeted more downstream pathways like TNF.
That's right.
Yeah. So is that in part what led you to believe that you should focus on an upstream pathway like IRhom2?
If you look at diseases like rheumatoid arthritis, for instance, I mean, there's these individual pathways that you're talking about that were clinically validated to be critical in the disease development, and as you mentioned, there is a TNF molecule, and there is IL-6 signaling, and well, both, and actually there's more pathways beyond those two, but these are the ones that were clinically validated. Those are also being regulated by TACE IRhom2, and well, we come from the oncology field, and
I didn't know that.
Yeah,
right,
so the company that I was with before, U3 Pharma, that was focusing on antibody development in the oncology field. And in oncology, I mean, it is standard to target different pathways at one and the same time in order to avoid the tumor to escape the mode of action. And well, that was a concept that we thought could or should maybe also be translated into autoimmune diseases, because all the single pathway inhibitors, they, well, turned out to work quite well, but there were limits. And it was just the concept to target multiple pathways at the same time in order, well, to hopefully increase efficacy. So that was the idea, and that made us believe that targeting a regulator that drives, for instance, these two clinically validated pathways and several others on top should be a good approach to improve efficacy in the clinics.
Yeah, absolutely. And what point then did you join the party? Yeah.
Oh, I joined the party when the company was already, let me quickly count, 6 years old.
Okay. So
Um,
where was the lead
it,
asset at that
it,
point?
it was basically almost ready for the clinic. One need to mention that what Jens describes here sounds like a piece of cake.
I can imagine
Just
it's
think
not.
about it and then develop the antibody and then let's go. But it was obviously, and your colleagues were, I'm sure you thought at some point in time, your former colleagues were right, that it was a crazy idea because it was quite a challenge to get to the antibodies.
Yeah.
And that's also why it took already 6 years before then I joined and they were at the stage close to enter the clinic.
So 6 years is a long time to have that level of conviction.
Tells you something
It's
about
impressive,
that guy,
yeah.
huh?
Yeah, I
There's an
mean,
entrepreneurial toughness to that, I think.
I think you have to be convinced about the
Yeah,
idea.
yeah.
Otherwise, you would never do that. Because honestly, I think, and I have a huge respect for those guys, because you basically start with something completely new, which is only existing in your mind and on paper.
Mm.
And you have to believe in that to not give easily up. And I think that is something which really is, I personally find quite an excellent
Yeah,
work
inspiring.
ethics. And that's something you need to have to, in order to go for these kind of things.
Is that something that the AI revolution with the silico trials and stuff, is that the difference between thinking of it in your mind and on paper to actually doing it? There's a lot in between that. Is there anything that in silico trials and things like that could do in between that to help the world of biotech get to the end result more productively, or are we still a long way from that, just out of interest?
I think one of the big problems, at least always for me, is we know quite a lot about the human body and what works inside, and with having deciphered the human genome, etc. So we have made quite a way forward. But we still, I believe, underestimate the complexity of the human nature,
Mm.
especially if you think about the immune system. I mean, the world around us is not a friendly one, to be quite honest. I really mean the things like bacteria, viruses,
Yeah,
all of
yeah.
that stuff. And I mean, we need to have a very, very complex system to constantly fight that back in order to survive.
We just had actually, where in my town, 6 cases of meningitis for the first
Yeah,
time.
I read that. Yeah.
Yeah, and that's where I live, and that's come out of nowhere, right? That's been
Yeah.
everyone's been talking about it for a long time.
Yeah.
So I see what you mean. I think people just living in close proximity and things like that.
Exactly. And unfortunately, the way we live over the last couple of years probably has led to the fact that we, in many cases, do not train our immune system enough. And in other cases, um, probably have, again, by the way we live, have led to the reoccurrence of some viruses which were there before, then were kind of controlled for a while and then came back now. And I mean, we have a couple of those cases. You mentioned the meningitis. Does not necessarily have to be virus. There's also a bacterial form of that one.
Mm.
By the way, I suffered from that one when I was a small kid, meningitis.
Oh.
It's not fun, I can
My
tell you.
business partner did the same.
Yeah.
Same thing, yeah.
So, but again, I think we always underestimate the complexity of the human nature and of the immune system. So we believe we have deciphered it. We believe this now has to be the key. It must work. But then we try it, and then works to a certain degree, and that's what Jens also mentioned to many of the drugs out there, the TNF blockers,
Yeah.
the R6 receptor blockers, great drugs, no question. But they get to a certain level of efficacy, and then they hit the ceiling. That's it. And we have a strong belief that the key here might really be the combination. It's the monopathways which get you to a certain level of efficacy. But if you really want to get to the next level, you need to hit more than one. Now, that was tried in the very beginning. Once the TNF blockers came out, there were some early trials of a combination. I think there was one TNF blocker with an IL-1 blocker, for example. had to be stopped because of serious side effects. Serious infections got up quite a bit. So that then led to the belief that in immunology, please do not combine these anti-inflammatory drugs because it brings down your immune system too much and then you are prone to these infections. Now, I think we have gone beyond that in our approach, definitely, but also in other more clever combinations. There's a couple of trials being done of combinations for certain monopathway inhibitors, which have led to increased efficacy without the burden of these increase in serious infections. So again, we are learning. We see what we may be able to combine and what not. And I think the trick here for our mechanism is the way we combine it. So it would be wrong if you would think, okay, targeting IRhom2 is basically, and I restrict it now to the two pathways we already mentioned, the TNF and the IL-6 pathway, is just a simple combination of the two. Because that was a question we were faced with from some of the investors when we were trying to get our Series A round together. They say, okay, both drugs are there, TNF blockers, IL-6 blockers. People could simply combine it. What would be the advantage of you guys then? But I think there you have to go one level deeper. It's the way how we target those pathways.
Well, it drives a much higher level of efficacy.
Yes, by not increasing the safety risk.
Well, yeah, okay. And what's out of interest? What's the general increase in level of efficacy?
You can't predict that.
Have you got
I
like
mean,
a
we
rough-
have preclinical data
an
which
estimate?
clearly show a very interesting and high increase in the efficacy, even to a degree in an IBD model, preclinical IBD model, that we saw not only stabilizing the disease, but getting better despite continuous challenge. So you have something where the burden for the animals was on a constant decline, basically. So you could speculate that if you would have continued this study, which for those studies at some point there's an end just because of the lifespan of the animals, they would have been cured, basically. Now, cure is always a very high word, and I would never
Yeah.
claim that we cure the disease.
It's not as black and white as that.
Yeah.
typically.
But clearly, I mean, that demonstrated that we have a very high level of efficacy to expect. And now the question is, does that translate into humans? Also there, unfortunately, in the past, in the drug development history, not of our company, but in general, there were quite some surprises where you had effects of a new mechanism which were quite astonishing in several animal models, and then they went into humans and flat nothing. So that's always the question. That's why you have to jump into the clinic at some point in time and test how it really works in humans.
You've obviously done something right in the way that you articulate that message, and it's again comes down to the conviction, because you raised a really successful Series A. Congratulations on that, by the way. So what do you think it is about your program and product and your lead compound that is attracting investor attention over other?
I think it's probably a couple of things. Certainly, on the one hand side, it's the novelty of the mechanism of action. I mean, that is what is in many cases driving industry, going beyond the VCs, thinking about big pharma. It is the promise of that high efficacy. And thirdly, it's the promise of a pipeline in a molecule. Because if you think about it, I mean, I think the TNF blockers, Humira, in the end
Yeah, I was
was
thinking that,
approved
yeah.
for 12, 14 indications or so. The IL-6 receptor blocker, tocilizumab, is also approved for 5 or 6 indications. In theory, you could make up the math and say, okay, if both work, why should this molecule, the IRhom2 antibody, not work in all of those indications
Yeah.
as well?
So what had
And I think it's also the fact that we found a new way of targeting TACE, which, as I mentioned earlier, it used to be a very popular target, and there was probably good reason why all the big companies, the big pharma companies, try to find a molecule to interfere with TACE activity. Yes, that may have caused some hesitance by one or the other investor who may have been aware of those previous failures, and we had to convince them that our approach is a different one. But those who believed in the potency of TACE inhibition, and those that we could convince that targeting TACE through IRhom2 is a different approach. I think they shared our enthusiasm and also the belief in this molecule.
Yeah, it's a good combination of a compelling, different scientific narrative with value proposition, with solid funding, with some really institutional backers and people that have been in the industry before. So, so it's very exciting. Can you tell us more about the lead program for the audience?
Yeah, sure. The lead program, the molecule is called SR878. As you know, they always have these funny abbreviations in pharma. It is in the clinic now. We are about to start a phase 1 in RA patients, actually. So that first patient in probably in the next couple of weeks.
Keeping yoga
Should
busy.
be pretty, yeah, absolutely. Absolutely.
Yeah.
And what we hope, I mean, in addition, you know that phase 1 is usually about safety, about understanding the PK, etc. But we also hope to get some good PD biomarker data out of that one. And the good thing is, by going into patients now, we can see effect on those biomarkers as well. Some of the biomarkers you would usually go for, I mean, we talked about that it influences the TNF pathway, so it's logical to look for TNF. You would not be able to do that in healthy volunteers because TNF is not increased, and so it would not react. So that's the advantage of going into RA patients now.
With any anti-inflammatory indication, I imagine there's challenges about trying to identify which is the best one to go for or optimal one to go for. How did you go around that difficult decision?
It was quite a discussion because, I mean, if you usually talk with investors or also with pharma guys about the indication you're targeting and you mention RA, there's usually some hesitation around that.
Why do you think that is?
It's quite an interesting thing. I mean, it is a huge market. I think currently sales are around 30 billion worldwide. But people believe that there's quite a number of drugs available out there. And probably due to the fact that they all hit that ceiling of efficacy, because that's the typical market where you can really see that. All of them, whether you take the JAK inhibitors to take the newest class there, if you take the older ones, the TNF blockers, the IL-6 blockers, they almost all basically hit the same amount of efficacy. But nobody has been able to go beyond. So it is a mix of, there's lots of drugs available. First ones, TNF blockers, for example, already going out of patent. So it's becoming genericized to a certain degree. And this experience that you have not been able to go through that ceiling, that people say, it's a tricky market. It's huge, yes, but if you want to get a interesting chunk there, we don't know how easy it will be to really get there. So that's why people kind of shy back a bit from that market. We actually believe it is exactly the right market specifically for our mechanism. Why is that the case? Because as we said, the TNF blockers and the IL-6 blockers, they are established. They are kind of standard of care there. So if we want to have an indication where we can prove that by hitting IRhom2, you're clearly superior than those individual pathway inhibitors, that's actually ideally then RA.
Because it's driven by not only these two clinically established pathways or clinically proven pathways, but there's also other pathways, other drivers of disease that are modulated by TACE IRhom2 activity and that we believe we can interfere with. So it's indeed the biology of RA that in many aspects is driven by TACE IRhom2, which we approach.
And you've done all of this and made all of this progress in probably the most turbulent biotech world that I can remember. So what's been your key to success in that world? You generated investment, you built a successful team.
Yeah, I think what one has to say there, in all fairness, it was tough to get to that, in the end, really very successful Series A financing round. We basically talked to more than 150 VCs.
Wow.
It took us 2 years to get there. So it was already tough at that time. We closed the Series A mid-24. But I'm actually quite happy that we don't have to be out on the road for a financing route right now, because I think it hasn't gone better. Quite the opposite.
Yeah.
There's some light at the end of the tunnel. It seems to be that also the IPO route is reopening again. There are a few new IPOs scheduled, I think, for this year. So let's keep the fingers crossed that that is happening.
I saw Immunic in the Munich kind of immunology world. They raised a successful round recently, didn't they?
Yeah. I mean, there are some
Promising, yeah.
examples, but there are also, I personally believe, quite a number of interesting scientific concepts which basically disappear because they don't get funding. And that's one of the problems we have. I mean, I can certainly also understand the financing side, the VCs. But I think that's a problem we have to get to these early funding rounds, seed, even pre-seed sometimes, and really get the support there in order to build up a package like we did, which then helps to get the larger VCs on board. Because you need to have some convincing data. Just to have the idea is good, and to have then people around like Jens who can, with a certain passion, talk about that mode of action is helping. But in the end, I mean, they also want to see some data. And if you are in the very early stage, you have ideas, but you don't have really data. And then to find people there supporting you, basically financing you, is not easy. I mean, we were basically before that Series A, we were basically supported by private investors. And we have to be very grateful to them because they really kept us afloat. I'd also like to mention the HTGF, the German governmental VC, so to say.
Who are the people that, I imagine there's probably partners in those funds that backed you from day
I
one?
mean, as I said, the majority were private people. But there were also the HTGF, and Frank Hensel is to mention there, definitely. He was a great support. He was one of the first ones who basically believed in us. That was even predates me. I mean, he was in before I even got on board.
Yeah.
And I think that also gave already a little.
What was the moment, sorry to interject there, what was the moment when there was probably a moment there where you knew you'd sold isn't the right word, but you'd articulated the vision, the mission, and the opportunity well enough. Was there a specific meeting where you think, oh, that was the time?
Well, I mean, definitely it was the first meeting with one of the private investors.
Yeah.
Which we had just, well, interestingly, just a couple of hours after my daughter was born.
Oh, congrats.
She was born just in
How old is she now?
in the night before. She's 9 years now.
Oh, my oldest is 9, yeah.
And so that was a memorable day because I
For many reasons,
basically
obviously.
left the delivery suite and then we went home, get a shower, and then we had our first private investor meeting. Oh, it obviously
It's a good icebreaker.
yeah. Well,
just had a baby.
It worked quite well. Right, just a couple of hours ago. Yeah, so that worked quite well.
She's not called SciRhom, is she? I'm joking. A good way to mark the day.
IRhom. No.
Yeah.
No, so that definitely that was a very, very good meeting. And then also the first meeting with HTGF when we presented, and well, people there who also believed in the concept, even though at that time we had nothing more than an idea, than a hypothesis. Then I think quite a good experience how to develop antibodies. And that was basically what they believed in. And so HTGF and all the private investors, they were the ones who believed in the idea, who, agreed on the potential if an antibody can be developed, and who supported us since then. Yeah, which was, I think, a great team of people who, from different perspectives, contributed to the program and to the progress of the program.
Yeah, amazing. It's a great story as well.
Yeah. And then, I mean, it comes in stages, actually, because as you know, drug development is a very costly endeavor. So it was clear that for the Series A, we needed VCs on board.
As in institutional ones.
Yes, the institutional investors then, basically.
Who's the lead in the current round?
It's interesting. We have 5 lead investors.
5 leads.
Yeah.
That's a busy boardroom.
But really, really good ones. We have
Yeah.
Andera from France. We have Kurma from France. We have Wellington partners here in Munich.
Yeah.
We have MIG funds, MIG Capital here in Munich as well. We have Hadean from the Nordics. And we have Bayern Capital, definitely. So
Who are the people that sit on the board from those funds?
all of them. So we have a relatively large board, seven people now. But it goes really well. I mean, we have good discussions in the board. We have Olivier Litzka from Andera. We have Peter Neubeck from Kurma. We have Veit Jan from MIG Capital. We have Varun Gupta from Wellington Partners. We have Georgina Askelon from Hadean. So really,
so how does it work that you have 5 leads?
Not to forget Natalie Weitemeyer. So just
sure,
to have
absolutely, from Bayern Capital.
The round
Full
complete
suite.
from Bayern Capital.
How does it work? I've never heard of that. There's 5 lead investors before.
Yeah, I mean, it kind of
You
evolved.
often hear the opposite. You hear
it kind of evolved. We had, and credits definitely to them. We had Wellington Partners throwing in the first term sheet, but then it became clear that there's more interest from other VCs who wanted to jump on board. And actually, one also has to say that the original planned Series A we wanted to have was clearly smaller than what we ended up. So there was oversubscribed, and there was quite some interest.
Congrats on that. That's a big thing in that climate, right?
I mean, it was, as I said, it was a long and tough journey, but in the end, it was clearly very fulfilling.
Very London bus syndrome. I don't know if you've heard that term before.
No.
You wait for one, and none comes, and then 5 come at once. That's and it feels like that when you describe it.
See, that's we should.
2 years waiting for the bus, and then you had 6 at once.
Yeah, exactly. Yeah, and then in the end, we agreed that they will all be lead investors. And now we have all of them on board, and it's really working pretty well. Yeah.
Exciting. So what's the next big milestone that's in everyone's minds as to where
the next big milestone is clearly getting the data from that phase 1 study in RA patients now. That's decisive in many instances.
What's the timescales targeted?
The trial, since it is in RA patients and it takes longer to recruit there, so we do expect to have the data by mid of next year. So a bit more than a year for the trial itself. It's a single dose trial, escalating, ascending doses. So it will be very interesting.
What does a win look like in that first trial, do you think? What's in your mind?
I'd like to answer that kind of nuanced. There's a minimum win, which is adequate safety, that nothing pops up on the safety side. I mean, that's what the phase 1 trial is usually for. The second kind of level is to see biomarkers going in the right direction. There's certain IRhom2 TACE substrates which we could measure and see whether they go into the right direction. That could be up or down. That's why I make this one, depending on the substrate and how you measure it. And certainly, I mean, but that will be an overwin already. However, I'm always cautious because we're talking about very small patient numbers. A clinical signal would be wonderful. There are numerous examples in pharma where you saw a very early signal in a phase 1, which you were then not able to reproduce. I mean, that's biologic variability in humans, and it may be that you see something in one patient or so, and it's not reproducible. So would be great to see something, especially if you can correlate it then with the biomarkers, that you see the biomarkers are going into a good direction and the clinical signal is then also there, because then you have a correlate. If you would only see a clinical signal without the biomarkers moving, somehow suspicious. But that again would be the super exciting result. But even if it's not there, if you don't see a clinical signal, again, with those small sample sizes, I think you cannot expect that.
What it sounds like, though, is that you're not going to be a single asset company story, which has often been the typical biotech life cycle, more building a platform for expansion, like a Humira. Assuming you hit the required efficacy.
If you're talking about a pipeline in a molecule, absolutely. And there are also, I mean, Jens mentioned that there is lots of indication opportunities even outside of the autoimmune field.
Oh, really?
Yeah.
Any in particular? Or?
I mean, you can think about oncology, various oncology indications. Now, coming back to our current molecule, you probably don't want to have an antibody which brings down TNF. There's a reason why it's called tumor necrosis factor. So, but interestingly, there may be possibilities to target IRhom2 and do not touch on the TNF pathway. So there are opportunities there to go for other antibodies with a different profile. So in addition to just having a pipeline in a molecule, there may be also a molecule pipeline to turn it around.
Yeah. And I suppose there's also the risk of going too broad to start with, right, when you're managing resources effectively. How have you struck that balance between speed, capital, talent? Any kind of secret sources you've juggled along the way that you think
Yeah, speed is usually a thing which you more or less automatically
live by
have in biotech.
Yeah.
Simply because you have a much smaller organization and your steps of getting to a certain decision is a lot easier than at a big pharma company. However, that is usually also constrained by capital money because that's the scarce resource in biotech as well. So it's basically a permanent balance, trying to keep the foot on the gas pedal as much as possible, but in keeping in mind then also the capital resources you have.
And I mean, it's also to, pay sufficient attention to the science. We strongly believe that science and a solid science, solid development activities are key. So, for instance, I mean, it's again a balance of effort and time, but it may be worth to spend a few more weeks on, for instance, or maybe even months, on, for instance, the identification of the lead candidate, then picking a potential lead candidate too early, moving ahead with the next steps, which may be very costly, which in the case of an antibody are extremely costly. So if you think of the CMC development, for instance, that's a very, very expensive journey. So we always try to balance time and money and solid data that we need to be convinced about in order to take the next steps. And this is something that was also shared by our investors, who, of course, also, as much as we were, interested to drive the project as quick as possible, but also on a solid decision basis. And I think that's something that we apply to all the questions, the critical questions that we try to answer, regardless of whether or which one is the lead candidate to finally develop, what is the best strategy, also in terms of disease selection. So we've spent quite a large amount of time on the selection of the diseases that we want to go after first. So solid science, solid data packages is something that we feel is key.
Great people, right?
Absolutely.
Yeah.
Absolutely.
Everyone's very experienced.
And that's something that we can't that is very important, that can't be overestimated enough. So we have got a very, very good team, people that we know, some of them for 20 years. Some of them worked with us even in the previous company. Then our CSO, Matthias Schneider, has a lot of experience in antibody development. He also was with U3. Then input from Carl Blobel. So talented people definitely is key.
And I think what you also have to always keep in mind, there's a temptation once you have closed a successful round, like we did with the Series A, that you increase the size of the company. I think you have to do that very carefully. Because again, that is basically what burns quite a lot of money. And you need to always judge on, do I really need that additional position now? Or can I still go with external support? And I think that is something you have to balance quite carefully, really, in order to concentrate further on the drug development and not lose yourself into building up a large organization.
So I'm very fascinated by the founder dynamic, and obviously you came on as a CEO later in
Mhm.
in the company's development. So how do you break up decision making? How do you split that between you, Jan, and the rest of the founding team? How does that whole thing work?
I think the fundamental topic here is that I personally, and I think the rest of the management team as well, we believe that it is actually a team exercise. I don't think that I'm the only one who should and needs to take decisions. I think for me, it is important to involve the management team. Usually, more than one mind can think more than one mind, and brings in different aspects. Sometimes we have tough discussions about various topics, and we
Football?
are not. Football is one thing, but
Yeah.
that is always a difficult one.
Yeah, I'm a Chelsea fan, so I'm struggling at the moment.
But also with regard to the next steps within the company, what to do, how to do it, we do have quite some controversial discussions. But the good thing is really to have the discussions, and then in the end, come to a decision. And then everybody feels, okay, we have discussed it, and we have found and identified the best way forward.
Yeah.
And I think that's important. It's again, I'm clearly not a fan of I'm the boss and I decide everything. There's quite a couple of topics where honestly I don't have a big clue, so I shouldn't decide on those. I do think I have a certain knowledge about the drug development and how the pharma market functions. So I'll bring in that expertise, but there's other expertise around the table, and there it's important to really get this expertise on the table and then come to a decision. I mean, there are certainly some areas where I primarily focus in. There are other areas where Jens is primarily focusing on, for example, the CMC part, so the development of the antibody production
Yep.
process and
Mhm.
all of that. He has started that. He has the expertise that clearly stays with him. No doubt about that. I mean, if there are topics to discuss, we again discuss it. But in the end, it's mainly Jens taking the
There's
decision.
definitely a very effectively democratic flavour in the company,
Yeah, absolutely.
even from the investors, if you think about it.
Yeah.
To have like several different leads,
Yeah.
which is really impressive, I think, when I compare it to biotechs that I work with. Usually it is more. It starts off not top down, but it becomes very top down.
Mm.
So,
And I mean, one should keep in mind we are a very small company. We're just, well, two hands full of people. Well, we basically, I mean, we share the same offices. I mean, we do see each other every day. So
I always say this, we spend more time together than we do with our families,
it's true.
with our business partners and stuff like that.
Yeah, it's a bit of a sad fact, but
Yeah.
unfortunately,
Although I
it
don't
is
think my
true.
kids would find it very interesting if they came to work with me.
Yeah, mine are old enough. Mine are
Yeah.
30 and 32. They have their own business. So
Yeah. I don't think a 5, a 7, and a 9-year-old would have a great time coming to Munich with me. Well, maybe they would. But they'd
yeah,
definitely be
where are they,
distracted.
by the way?
Yeah. So based on what you just said, Jan, coming in after the founding,
Mhm.
how did you manage that dynamic, and what were the biggest gaps that you came in to address?
Say the right thing now.
There were no gaps.
No,
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 no.
that's what I.
Yeah.
No. Basically, I mean, I think
What was the mandate, I suppose, is probably what it comes down to.
Yeah, I think what the board at that time was looking for, and that's why they approached me, is somebody who has done it, seen it, so and
Commercialised it.
commercialized it. Yes, and so bring all of that expertise into the discussion, and then define the best way forward. And I think that is basically what I tried to do. It's not that, for example, the idea of going into RA wasn't there before, but there was quite some discussion also with the board whether that's the right
Mhm.
decision or not. So we took another look at it, and then in the end said, no, it is actually the best decision to go into that. However, we expanded it a bit. We said, okay, why not? And that's probably not really typical for a biotech company. Why not, instead of just doing one phase 2A trial, do 2 of them in 2 different indications? So you do not put all your
Spread the
eggs in
risk,
one
yeah.
basket, and you clearly enhance also the profile. I mean, it does make a lot of a difference also for a pharma company, which at some point in time you would want to have somebody on board to support you, where it's really also then getting attractive.
So Jens, where did you, from an outside-in perspective, because Jan is obviously far too humble to answer this honestly, when Jan came on board, what was the biggest contribution he made?
Was there any?
Well, I mean, definitely it was his big pharma experience. I mean, and that's something, I mean, that we did not have in the founder team. I mean, we used to be with Daiichi Sankyo, U3 Pharma, that Matthias and myself was with before was taken over by Daiichi Sankyo. So we, of course, did have exposure to big pharma, but not from a developmental perspective. And this is something that Jan brought in. So, well, he was involved in the development of a very important and successful molecule, SciRhom's. And of course, that's an expertise that none of us had. And well, bringing in the expertise and the knowledge of developing such a successful molecule, of course, that was a rich value to the company.
As someone that's commercialized
Yeah.
before, you can make decisions at the earlier stages, despite it being a long way away, being in phase 1 at the moment. But you can make those decisions now with the end in mind.
Mhm.
So you have a clearer path as
Mhm.
opposed to kind of going zigzag.
Yeah, and I think that's an important aspect. And quite frankly, I think this is something which, at least a while ago, was also not very prominent in big pharma. I mean, there was a time when people, when thinking about developing drugs, thought about, okay, it came from research, they had their nice ideas, and then it was handed over to medicine, and medicine had an idea how to develop the drug. But nobody really looked at the question, okay, what profile does that drug have to have in order to be successful in the market 10 years down the road? Because we should not forget that's how long it takes to develop a drug. So you need to anticipate how the market looks like in 10 years. So what you need is a crystal ball, a big one,
You'll need that, don't we?
and really think about that from the beginning on. It is an evolving picture. So it's also not the case that you can, at start of the clinical development, sit down and write down how your SPC should look like, and then this doesn't change. I mean, there's a give and take, basically. You go in with a certain idea, what you want to do, and that is then reshaped either by what you get out of the trial or by what is happening out in the market. So it's a dynamic, basically. But you have to think on early what you want to develop so that you can do the right trials. And I think that's important to really keep that kind of commercial perspective and bring that into the drug development process.
Yeah, it's interesting that you're saying that's lacking in big pharma at the moment.
Not at the moment anymore. I think they have developed quite nicely, but if I think back 15 years ago, it was a relatively novel approach.
I think the role of medical affairs is a good almost metaphor for what you're describing.
Mhm.
I remember traditionally when we were doing searches for medical affairs folk, they were more seen as, especially if they were physicians prior, they were more seen as the medical authority that sits in the background. Was now pretty much all the time now that one of the first things they'll say is that they want them to have that, but that needs to be complemented by a commercial mindset and someone that can liaise with the sales team to help really effectively market this drug ethically as well and bring the two things together. So it's quite interesting.
Yeah, I think that is definitely important. I mean, in the end, we definitely want to develop drug in order to help patients, but there's no way around. We also have to be commercially successful. Now, you can have a long discussion whether the prices are at a level where it's probably too high. I don't want to go into that discussion, but you need to earn money with what you're doing because otherwise you will not be able to develop further drugs. So I think, and there's nothing wrong in that. As long as you keep in mind that you want to help patients to better deal with their disease or in a best way, if possible, even cure the disease. That should be the ultimate goal always. But again, I think since it is a very costly endeavor to develop a drug, you also have to earn all of that money back.
So if everything goes right with our crystal ball, what is the what could this company become?
That's a very tricky question because if you think about, we mentioned that in the beginning that there's quite a number of indications you could go for. And I think what you cannot afford nowadays is develop one indication after the other. Bring it to the market with one indication and think about the next and so on. Because you're always running against your exclusivity period.
Yeah.
So
Just
you
trying
have
to
to do
develop
a
it.
lot of things in parallel, which in the end, however, does also mean that it is very costly. And if you then think about it, it can easily sum up to an amount of 1 billion, just the development. And that's an amount which is, for a small biotech,
It's eye-wateringly high to think about, isn't it?
hard to imagine. So I think at some stage, if you go for these kind of molecules, for a pipeline and a molecule, you have to go for a big pharma partner. Now, that doesn't mean that you have to completely stand back and say, okay, we sell it and we don't do anything with it. There's multiple models. And that's what you then have to evaluate for yourself together with your investors. What is it what you in the end want to do? Do you want to continue to also further develop SciRhom as a company? Or are you fine with completely exiting? I think that is something where I believe you should not too early decide definitely what you want to do. You have to create certain options. And I think that is what you will always have to do, and think about it, think it through, discuss the pros and cons within the team, with the investors, and then take a decision. And I mean, it certainly also depends what your options are. I mean, you may say, okay, it looks nice. We want to go now into the broad development, but you have to find somebody who's interested to join you.
But definitely, we want to be the IRhom company. So
That's true.
that's what the name already says. So SciRhom, that's, well, I mean, it picks up the target that we are addressing, well, I think that's definitely our goal. So whenever, from a therapeutic perspective, one talks or thinks about IRhom, in particular IRhom2, it needs to be associated
Yeah,
with
I like that.
SciRhom.
Yeah. And you
Yeah.
mentioned about the total cost of drug development can cost up to $1 billion.
Easily.
Yeah, we had on the podcast actually. We had a guy called Ed Cliphouse, who's the head of the partner leading Sophanova Digital, which is Sophanova's effectively their tech bio AI orientation, but is within life sciences. And interestingly, one of the main thing he talked about wasn't about the opportunity of improving the quality of drug development. It was about the very near term potential of creating cost collapse, and that is something that is close. When you talk about that and you think about the 9 out of 10 trials fail, kind of general rule of thumb, there's a massive opportunity there, isn't there, for tech bio to come in and strip away some of that. Although I appreciate it's easier said than done.
Exactly. I think that's the problem. And it comes back to the issue which we touched on earlier, and that's the complexity of the human biology. I think we should not underestimate that even with the features and the promise AI may give, I still have my doubt how far AI can really trim that really significantly down, especially for new modes of action. I mean, yes, IRhom2 is already in the human body for forever, probably, but it has never been targeted by a drug. So I wonder actually how AI should then come up with what may happen.
And so to finish us off, we've got time for one more question. What's the biggest risk that could derail this journey?
The biggest risk is definitely failure in the clinic. So if it turns out either that there is a huge safety concern for whatever reason, or that there is no efficacy, that's probably the end of the story. But I mean, that's the nature of clinical development. That's why we do the experiment. Hopefully we get the outcome we want to have, but at least we get to a definitive outcome and can then decide what to do based on that.
And on a granular level, as people that have journeyed into the biotech world, so you are comfortable with a certain level of risk, you're bought into the journey. What is the kind of shared value, if you like, or shared mindset, which you think is a common denominator across the SciRhom team?
I think basically advancing the science in order to help patients. I think that's the core. I mean, Jens mentioned that I was involved in the development of SCYRhom's, the IL-23 drug for psoriasis. That's where it started. And that was an experience really where you could see how much you can help patients. That, by the way, was an example where with the first trial, single dose, we went into patients and already there you knew that you have a drug because you cured the psoriasis basically for all of them with one shot. Cured. You made it disappear for a while at least. And I think that is something which definitely drives me to really be able to help patients, because I believe even in a disease where people, and we talked about that, like RA, where people think, okay, that's actually served, the patients suffer still quite a lot. I mean, even with all the drugs, there's probably up to 50% of patients who do not ever even get a 50% improvement of their symptoms.
So low, isn't it?
Yeah. And that's there for so many diseases.
What age does rheumatoid arthritis generally kick in?
Actually, not at that old age. I think there are different forms. There's one which already can strike you when you're really young. The majority of the kind of classic RA is, however, in the older age only.
Well, thank you so much for joining me on the podcast today and sharing the exciting story of SciRhom. I can't wait to keep seeing it develop.
Thank you
We
for
too,
having
and it
us
was
here.
quite a pleasure. Thank you.